Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Hey everyone, Dr. D here, and in this video we are going to be covering Chapter 10 from our Brock Biology of Microorganisms textbook. This chapter deals with viral genomics, diversity, and ecology. So let's go ahead and get started. So we had a review of viruses in the previous chapter, but now we're just going to delve into what makes them different. First of all, we're going to be looking at the size of the viral genome, and this can vary up to 1,000-fold from the smallest to the largest genomes. The smallest viruses being the Circovirus. This virus has a tiny genome of only about 1,750 bases, so it's 1.75 kilobases as a single strand non-segmented genome. So very tiny genome. The largest genome virus is the Pandoravirus, which has 2.5 megabase pairs, so 2.5 million bases for this virus, which is actually a larger genome than some bacteria, believe it or not. This is a virus that infects some marine amoeba. Typically, RNA genome viruses are smaller, have a smaller genome than DNA viruses. And this chart here summarizes different viruses based on the length or the size of their genomes. Remember, the Circovirus being only 1.75 kb, whereas the Pandoravirus 2.5 megabases here, which is larger than some bacterium and about half of the amount of DNA for an E. coli, believe it or not. And look here again, the smaller genome viruses tend to be RNA viruses, and the larger ones tend to be DNA genome viruses. And remember the Baltimore scheme for viruses. There were 7 classes of seven classes of viruses. 2 of them are called the DNA viruses, class 1 and class 2. Technically, class 7 is also a DNA virus. However, because it uses an RNA intermediary, it's going to be categorized under an RNA virus. So we'll get to that in a minute. But the DNA viruses are technically class 1 and class 2. Class 1 being the double-strand DNA viruses, and class 2 being the single-stranded positive or plus-strand DNA viruses. Here is a summary of all the different classes of the Baltimore system. You've got class 1 and 7 being the double-strand DNA viruses, class 2 being the single-strand plus-strand viruses, class 3 being the double-strand viruses, RNA viruses, the double-strand RNA viruses, class 4 being the single-strand plus sense viruses, class 5 being the single-strand negative sense viruses, and class 6 being the single-strand RNAs, but known as the retroviruses. These are the 7 groups here, and what you're seeing in figure 10.2 is how each of these different classes can undergo transcription in order to make mRNA, and how, because it's vitally important that all of these genomes be able to make mRNA, and that's because you want to synthesize proteins, right? So different classes get to the mRNA via different avenues with different mechanisms. So now let's talk a little bit about the RNA viruses. Remember, I said the RNA viruses include class 4, class 5, class 3, and technically class 6 as well, because that's a retrovirus. The RNA polymerases. The RNA viruses need RNA polymerases. The RNA virus must replicate in virion or encode RNA replicase. Remember this enzyme, RNA replicase? It's very important to know about RNA replicase. RNA replicase is an RNA-dependent RNA polymerase. That means it can read, it's a polymerase that can read a RNA template in order to synthesize an RNA product. So think about it, if you have an RNA genome and you want to copy that into more RNA genome, you're going to have an RNA replicase so that you can copy that RNA. Remember that animals do not possess such an enzyme. So class 4, these are the single-strand positive RNA viruses. You should know that, remember from chapter 8, positive sense single-strand RNA is the same sense as mRNA. So these genomes can be used as mRNA. Let me show you. Class 4, single-strand RNA positive virus. Look what it says. Their genome itself can be used directly as mRNA. Did you see that? Their genome can be directly used as mRNA. In class 5, these are called the single-strand negative sense RNA viruses. The RNA replicase, this RNA-dependent RNA polymerase, RNA replicase makes plus strand RNA, and it uses that as mRNA and template to make more of its genome, more of its negative strand genome. Class 3, these are your double-strand RNA genomes, has a similar replication as class 5. So again, replicase would make positive sense mRNA. You can use that as mRNA and as template for more of the double-strand genome. Class 6, these are the retroviruses. Remember that even though they have genomes of single-strand positive RNA, this single-strand positive RNA is first reverse transcribed with reverse transcriptase. This would be a RNA-dependent DNA polymerase. To make double-strand DNA, that double-strand DNA, remember from chapter 8, that double-strand DNA will then insert itself with the enzyme integrase into the genome of the new host, such as is done with HIV. And then there's class 7. Remember, this was the weird one. This is double-strand DNA, but with a gap in it. But this double-strand DNA replicates through an RNA intermediate. It also uses reverse transcriptase. That's a very strange bird. So here we have the hosts for the viruses. Only certain Baltimore classes infect certain phylogenies. So check this out. Class 1, the double-strand DNA viruses, are primarily prokaryotic viruses, but not exclusively primarily prokaryotic viruses. Class 4, these are the single-strand positive RNA viruses. These are primarily eukaryotic viruses. Fungi, it turns out, are only infected by class 3 and 4. Most class 1 and class 5 viruses have animal hosts instead of plants, but more class 2 viruses have plant hosts instead of animals. Isn't that interesting? Class 6, this is your retroviruses like HIV, infect only animals, whereas class 7 are more common in plants than in animals. And here you can see, based on the domain of life, archaea, bacteria, eukarya, you have different amount of diversity of viruses. So archaea has the least diversity of viral types that infect them, and that makes sense. I mean, they're living in extreme environments. Bacteria are a little bit more diverse in terms of what classes of on the Baltimore System of viruses infect them. And eukarya, these are the most diverse in terms of the myriad types of viruses that can infect the cells and the viral diversity there. And if you break the eukarya up into their kingdoms, the fungi, the plants, and the animals, and not the kingdom, but the group of protists, you'll notice that animals are the most diverse in terms of the types of fungi. This little guy jumped up here and he threw me right off. Gizmo came to join us. Hey, Gizmo. How are you doing, buddy? Here, if you sit on this side, you won't block the screen. It's always nice when you come say hi, Gizmo. So Gizmo came to say hi. So again, what does this show us? This shows us that animals are so diverse in the types of variety of viruses that can infect us. Look how many different types. So it's 1, 2, 3, 4, 5, 6, 7. So all 7 groups, all 7 classes on the Baltimore system can infect us. Right? Isn't that interesting? And if we take a look here, viruses are also very diverse in terms of their shape. Remember, viruses can be icosahedral, viruses can be helical, viruses can be what's known as complex, where they have multiple different shapes like icosahedral head, helical tail, tail fibers. Remember all that? So viruses can range from very simple looking to quite complex. Now, when a virus is undergoing the synthesis stage, this is where the virus is producing more of its proteins and more of its nucleic acid. There are what's known as early proteins, which are synthesized soon after infection, and then late proteins, which are synthesized later. The early ones, these are usually enzymes, things that the virus needs in order to reproduce more of itself, made in small amounts. These include nucleic acid polymerases, proteins that shut down the host transcription and translation, so we could focus on viral transcription and translation. And then later on, these are late proteins, which are synthesized later on. These include structural and assembly proteins, which are made in large amounts. So eventually, new virion are assembled and exit either by lysis or by budding. If it exits by lysis, remember, usually they don't have envelopes. If they exit by budding, that's how usually you end up with an envelope. So when did viruses appear? This is an active area of research. Viruses may have arisen after cells. Viruses may have been the first forms of life. They may have been pre-cellular. And viruses may have actually played an important transition from RNA to DNA. But one thing is for sure, why did viruses appear? Well, they are a mechanism to quickly move genes from cell to cell. How did viruses arise? How did DNA viruses arise? Likely as protection from ribonucleases. You should know that this idea of RNA world is one that it's an interesting idea, and that is that early in evolution, the genomic material was RNA based. Why? Because RNA is a storage of information, but it's also able to serve as an enzyme. Ribozymes can copy themselves, they can cleave things. Ribozymes have functionality to them. However, RNA is unstable. It's easily degraded. Why? Because of the hydroxyl group on the 2' carbon on ribose. So it's thought that viruses played an important role from the transition from genomes of RNA only to genomes of DNA. And why is that? Well, because to protect from enzymes that break down RNA, they may have been involved in the transition by developing the enzyme reverse transcriptase, which is absent in eukarya. This enzyme can convert RNA to DNA. DNA viruses then infect ancestors of cellular domains like archaea, bacteria, and eukarya, and convert The host cells then began to replicate DNA and converted their genomes from RNA to DNA. Why? Because DNA is more stable than RNA. This is fascinating. So according to these ideas here, viruses may have been intimately involved in our transition from an RNA genome to a DNA genome with the help of their enzyme reverse transcriptase, which is absent from eukaryotic cells and from any cells, prokaryotic cells as well. Very interesting stuff. Here's some more interesting stuff. This is called the microbial arms race, viral defense mechanisms of bacteria and archaea. So as I mentioned, there's these bacteriophages. These are viruses that infect bacteria. And these bacteriophages destroy the host cells, the bacterial cells. But there's like an arms race going on. For example, look at this. The bacteria produce restriction endonucleases. These are enzymes that chop up DNA, like the DNA of viruses. So the bacteriophage, such as the T4 bacteriophage, substitutes 5-hydroxylmethylcytosine for cytosine in the genome, rendering the restriction endonucleases useless. So some E. coli evolved altered restriction enzymes that can degrade that modified T4 DNA. So the T4 phages modified the DNA again, but this time by glycosylation and not by 5-hydroxymethylcytosine. So what did the E. coli do? Some E. coli evolved altered restriction enzymes that could recognize and degrade glycosylated T4 DNA. So what did the T4 do? Some T4 inhibit these modified restriction enzymes. And then the E. coli, you know, in this arms race, some E. coli evolved uninhibited endonucleases. It really is an arms race here. So again, during the arms race, dynamic relationship between bacteriophages and hosts. So then alterations in viral receptor sites occurred. So what the bacteria did is it modified the receptor it modified its receptors, the receptors that are recognized by the spike proteins. Hey, Gizmo. Gizmo's come visit again. Howdy. I'll put you over here for now, Gizmo. What did the bacteria do? The bacteria mutated the receptors that are recognized by the virus. And the bacteria would shield itself with a capsule. So what does the virus do? The virus mutates so it can still bind to the mutated receptor, the modified receptor, or it also evolved to degrade the capsule, like use a capsule degrading enzyme or something. Some temperate bacteriophages can hijack the host's toxin antitoxin system. This one's fascinating. So some of these bacteriophages evolved to hijack the host's antitoxin system. This is the system keeping the bacteria safe from its own toxin. This is that fascinating. And Gizmo's back blocking the screen, so I can't really see what all's going on. Hi Gizmo. Hi buddy. Hi buddy. Yes. Okay. Here's Gizmo, and he's off. All right, this one is really interesting. This is CRISPR. I know it's pretty hot in the news. You guys may have heard about CRISPR used as a gene editing technique for embryo gene editing. However, its natural role where it was discovered was here in bacterial cells. It's an antiviral system of bacteria and archaea called CRISPR. CRISPR, by the way, is short for clustered, regularly interspersed short palindromic repeats. This is the system. It's like a very crude, basic immune system of bacterial cells that protect from bacteriophage infection. So you have regions contain short repeats of constant DNA sequences alternating with short variable spacers corresponding to memory of viral or other foreign DNA. So here's kind of how it works, and this is a basic rundown of how it works. So you have this is a bacterial cell, right? This is a bacterial cell. Here's your phage. Here's a bacteriophage. And what does a bacteriophage do? it injects its DNA. It injects its genomic material. Now, if the genomic material has a specific sequence on it called the PAM sequence, which is pretty common on the genomes of viruses, this PAM sequence triggers this enzyme called the memorizing complex of Cas proteins. This enzyme, when it recognizes the PAM sequence, it will actually remove the DNA adjacent to it called the protospacer. It will remove this DNA next to that sequence, right? This is called the protospacer. It then grabs that protospacer and takes it to its genome. In its genome, it's got these are the repeats, right? It's got a repeat spacer, repeat spacer, repeat spacer, repeat spacer. And what it does is it takes this DNA, this is the protospacer, the new spacer, and it inserts that into between the repeats. Does that make sense? So now it's incorporated that genomic DNA from the virus. It's incorporated it, and this all is part of the immunization part of the Crispr system. So this is how it gains the memory for next time. So then let's look at this part. This is the interference. This is what happens upon subsequent infection. There's another virus later down the road. This virus infects the cell. So here's the viral genome that's similar or the same as this viral genome. And what's happened? So transcription and processing of CRISPR RNA. So what happens is you have transcription of this CRISPR DNA into mRNA, and then you remove the spacers. The spacer then joins up with the Cas protein, and what happens is binding of the CRISPR RNA to viral DNA and cleavage of the duplex. You see this? The spacer recognizes itself. The spacer recognizes itself on the viral DNA, and the Cas complex then triggers cleavage of the viral DNA. So destroying the viral DNA. Isn't that fascinating? And so what people have done, if you've ever heard of CRISPR in the news, is they use this system to not cleave, viral DNA for bacteria, but they can edit this however they wish to target your own genome in your cells and modify your genome in various ways for gene editing. So we've kind of we've taken this system from bacteria and we're using it to modify genomes of animals and plants. Isn't that interesting? So again, CRISPR works with the CRISPR proteins, but also these Cas proteins. Cas proteins are CRISPR-associated proteins. That's what it's short for, Cas. These have endonuclease activity, so you see they can cleave DNA. That's what endonuclease activity means. They mediate defense against foreign DNA, and they can incorporate new spacer regions. That's what this is. They can cleave foreign DNA, incorporate new spacer regions. When virus attaches to the host cell, Cas proteins may recognize that PAM sequence, remember the protospacer adjacent motif. So they can recognize the PAM. If they recognize the PAM DNA sequence, then Cas cleaves the viral DNA near PAM, inserts the short DNA into CRISPR region as a spacer. That's what I was showing you here. It cleaves the protospacer and inserts it as a new spacer between the repeats. Cas cleaves the viral DNA near PAM, inserts, yes. This immunizes the cell. Then regions contain short repeats of constant DNA sequences alternating with short variable spacers corresponding to memory of viral or foreign DNA. So we talked about all that. When the immunized cell encounters the same virus, this is what we showed you on the right side of that image, Cas proteins destroy the incoming DNA. The pre-CRISPR RNA or pre-crRNA is transcript of CRISPR region containing RNA sequences complementary to the repeats of spacers. We covered all this. It's pretty interesting. So interference was the right side of this image. The interference is the right side of this image. It destroys the viral genome. So here's some questions. How does the host survive the initial invasion long enough to be immunized in the first place? Like when, you know, how did the host survive long enough to even get immunized on the left side of this image? Probably occurs when incoming virus is inactivated by the environment. So the virus luckily just died, you know, or got inactivated during the infection process. Or when restriction enzymes may have cleaved other parts of its genome. Viruses may have evolved. Viruses have evolved to avoid CRISPR over time. No surprise there. So, for example, just mutate your PAM region. Look, if the PAM region is triggering immunization, well, just mutate your PAM region. Don't have any PAM regions. That makes sense. Production of Cas inhibitors. So viruses have Cas inhibitors now. Phage encoded CRISPR in a phage that infects Vibrio cholera. So look at this. The phage encodes a CRISPR, and that CRISPR goes after the Vibrio cholera, which is just fascinating. This, again, it's this arms race that's going on between the viruses and the bacteria. So moving on to other agents. These are small infectious agents that are even simpler than viruses, but they can be spread. They're communicable. So these are called subviral agents. Viroids and prions. Let's talk about these. Viroids are infectious RNA molecules that lack a protein component. So think about a virus, but without the protein part, without the protein part, without the capsid part. Similar to virusoids. Remember hepatitis D virus is a virusoid. It's a piece of RNA with no DNA, with no protein component. Well, this is similar. It's got no protein component. Small circular single-strand RNA molecules, smallest known pathogens, just a couple hundred base pairs. Cause a number of important plant diseases, do not infect animals or microorganisms. So here you can see a healthy tomato plant on the left and a sick tomato plant on the right. This tomato plant is infected with the viroid. So what happens? The viroid forms hairpin-shaped double-stranded molecule with closed ends. I think I showed you here's a picture. This is a small RNA double strand because it kind of folded back on itself with closed ends. This shape that it adopts provides stability outside the host cell. When it can enter the plant through a wound. You know, obviously this thing doesn't have a capsid or spike proteins, so the only way to get this guy into a plant is through a wound. So for example, if an insect you know, introduces it or destroys or makes a hole in the wood, like termites or something, mechanical damage, some kind of damage in the plant, well, then that you could introduce this viroid. Then the viroid can move between the cells through the plasmodesmata. These are thin cytoplasmic strands. So you can see here, once the double strand, this orange stuff, the double strand RNA is inside, it can kind of move from cell to cell through these tiny holes, which are called plasmodesmata in plant cells. Completely dependent on plant RNA polymerases. Now, what's interesting is plants do have RNA polymerases, RNA-dependent RNA polymerases, and so they can actually copy these for the viroid. Let's see. So viroid movement inside plants. So yeah, once the viroid gets into the plant, it can move from plasmodesmata, from plant cell to plant cell, and it can also move through the plant vascular system, you know, the xylem and the phloem, to move to other parts of the plant as well. Plants may be asymptomatic or show mild to lethal symptoms. Most symptoms are growth related, so usually it's like the shape of the leaf or the shape of the plant in general. Viroids may mimic or interfere with plant small regulatory RNA. They're known to yield small interfering RNAs or siRNAs during replication, which may silence or suppress plant genes, specifically ones with leaf shape. All right, so moving on to our last concept. This is the last concept of the chapter. These are prions. Prions are like viruses, but without the DNA or RNA component. So imagine a virus, but without the nucleic acid part. These are just called infectious proteins. Prions are infectious proteins whose extracellular form contains no nucleic acid at all. And I'll tell you how these work in a minute, but they're very, very interesting. It's just a protein. And you come into contact with this protein and you ingest it or you get it into your blood and you could come down with these diseases. And these are horrible, horrible diseases. So they're known to cause diseases in animals, including transmissible spongiform encephalopathies such as Scrapie, Bovine Spongiform Encephalopathy, which is called mad cow, chronic wasting disease, Kuru, Creutzfeldt-Jakob Disease. These are not diseases that you want to get. These are diseases that cause spongiform encephalopathy, which means turns your brain into a sponge, essentially, by causing massive brain damage. Okay, let me show you a quick picture here. This image at the top shows you these massive holes that form in your brain. This is a section through brain tissue of a human with variant Creutzfeldt-Jakob disease. Note, this should say note the spongy nature of the tissue. You know, this looks like a sponge. You've got these giant holes that should not be in a normal brain, where neural tissue has been lost. This causes massive brain damage and death. So how does it work? Here's the mechanism. You have these brain proteins called prion proteins. You know, this is the normal prion protein, the PRPC, normal prion protein. These are proteins of the neural tissue and the brain, right? So here's the problem. If you encounter prion protein that has Scrapie, for instance, or misfolded prion protein is called PRPSC. So let me zoom in on this. Look, this is the prion protein that's misfolded. Let's say you got this in your diet somehow by eating brain tissue, or you got it in a bad blood transfusion or something like that. You've got this misfolded prion protein. A misfolded prion protein, and usually misfolded prion protein is caused by a mutation in the prion protein. So you've got this amino acid sequence that's a little bit different that causes the misfolding to occur. But anyway, you've got the misfolded prion proteins, then you've got your own prion proteins. But here's the problem. When your own prion proteins encounter a misfolded prion protein, this promotes misfolding of your own prion proteins. So you see what happens? It's like a cascading event. Your own prion proteins touch misfolded prion proteins, and then that causes your own proteins to misfold, and then that causes them to group up together, like clump up together. See, normally your prion proteins should not clump up. Misfolded prion proteins clump up, and these cause these aggregates in the brain. So you could see here accumulation and aggregation of these prion proteins form insoluble amyloids, which destroys the brain and other nervous tissue. Other amyloid diseases include Alzheimer's, Huntington's, Parkinson's, type 2 diabetes. These may or may not be prion diseases, but they end up having effects similar to these prion diseases. All right, so very, very interesting stuff. Prions are very scary, and there's a very, very, very low survivability to prion diseases, close to zero. Very, very terrible way to go. So I hope you learned something very interesting, chapter 10. As always, please leave any questions you have in the comment box below, and I will catch you in the next chapter.
Dr. D, 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 Dr. D. Dr. D.